Welcome to our first webinar in our new webinar series for this year, where we'll be looking at missing data and analyzing missing data and different ways to approach it in a clinical trial setting. So today I'll be talking about and giving an overview of missing data, the causes and treatments in clinical trials. So the format for this webinar is you can ask questions through the text box in the GoToMeeting window whenever you feel, and we'll be taking short breaks to address any questions as they come in. We'll also, if we don't get around to everybody because there's quite a large attendance today, we will be following up one-to-one with emails of any questions we don't get to or any discussions you'd like to have with us subsequently. So just to go through the webinar series first, there's 3 webinars. Today is the first one, where there's an overview of missing data causes and treatments in clinical trials. Next month in April, we'll be talking about smart choices for imputation in clinical trials and how to optimize your missing data analysis using imputation and multi-imputation in the clinical trial setting. And then our third webinar in May will be talking about sensitivity analysis and talking around successful strategies towards giving sensitivity analysis on your missing data for regulatory discussions and various applications in the clinical trial setting. So again, just to clarify, if the GoToMeeting window is too small, you can use the Zoom features there on the top left-hand corner. And again, feel free to put a comment through the text box if you're having trouble hearing or seeing the images or anything. So to get started then, just an overview of what I'll be talking about today. I'll be talking about some reviews of missing data analysis in clinical trials to date, various reports that are out there in the public domain at the moment. I'll give you an overview of some causes and examples of missing data in the clinical trial setting, such as reasons for dropout, impact of dropout in clinical trials, and also various steps to minimize dropout. We'll be looking at getting to grips with your missing data once you've collected your data at the end of the trial, getting ready to tackle missing data analysis then, with a view of imputation down the line or any other applications you have, and then we'll make some time for questions and comments along the way as well as at the end. So just to go through a review of what's been out there at the moment. So we're going to focus our comments and talking on 5 major reports that have been out there in the last 10 to 15 years. One of the main ones out there, of course, is the International Conference on Harmonization E9 technical report entitled Note for Guidance on Statistical Principles in Clinical Trials, and in specific Chapter 5 there with the statistical analysis guidelines on it. In that chapter there is a specific section on missing data analysis and approaching missing data. There was a report recently from the Office for Human Research Protections entitled Guidance on Important Considerations for When Participation of Human Subjects in Research is Discontinued, and that was published about, it was a draft that I published I think about 4 or 5 years ago at this stage. There was an excellent report released by the National Research Council. Sorry, the logo is mixed up here, but the National Research Council is third on the list there, and that's actually the bottom logo there, sorry, is entitled The Prevention and Treatment of Missing Data in Clinical Trials, and they were asked to do up that, there was a panel created and they were asked to do up a report on approaching missing data in clinical trials by the FDA, as they are ongoingly and actively pursuing guidance on this topic, because it is quite a prevalent topic in clinical trials. Also, the FDA themselves have a report out there called Guidance for Sponsors, Clinical Investigators and IRBs, Data Retention when Subjects Withdraw from FDA-Regulated Clinical Trials. That was published again about 4 or 5 years ago. And then the EMA, the European Medicines Agency, have a report called The Guidance on Missing Data in Confirmatory Clinical Trials, which was published about 2 or 3 years ago in 2010. So these are all great resources for getting a feel for what's out there at the moment in terms of thoughts and approaches to missing data analysis in this setting. So just some summary points and opinions that I was picking up from these reports are I think they all acknowledge that it's inevitable the occurrence of missing data throughout the clinical trial period, and that I think nobody expects there to be 100% data in many situations, especially in phase 3 trials, because it can so easily occur. But everyone seems to be on board that ignoring this missing data does bring up issues, and especially the EMA were quick to outline there that it violates the intent to treat principle, where data, even from those who withdraw from trials, must be kept within the study. So I think all organizations here as well kind of acknowledge that missing data is always a potential source of bias and loss in power and sample size from the trial, and can affect your inference and can affect the way you interpret the results from any analysis and any subsequent analysis that you do on the data that you have. And also the missing data can affect this randomized sample. So if you started off with a randomized clinical trial, because you've got some people withdrawing for whatever reason, the randomization may be reduced there, and there could be some issues creeping into the data and some bias there. Not in mind, if you do ignore these people, you are reducing your sample size, you are reducing the power then of your ability to pick up any real results. So I think they all were coming down on this message that there isn't just one approach out there to treat missing data or to approach it, but they feel that acceptable approaches will depend on the assumptions that you make when you're doing it, and whether it was reasonable to make those assumptions is an important aspect that needs to be clarified within the reporting and within whatever discussions you're having with regulatory agencies. But I think they are willing to get into discussions about this because it isn't as well defined, and there are slight grey areas. But at conferences and through these webinars, a lot of the people I've been coming across and the people I've been talking to on this topic, they've all been quite positive about imputation especially, and also that they've all been encouraged by several agencies to approach imputation as a viable method to deal with missing data. Also, I think a lot of them were picking up on this sensitivity analysis should be used at the end. It's kind of like a last step and more of a validation that you're able to then stand over your assumptions be made, and this is more of a kind of a solid approach to being able to stand over your analysis, the decisions you've made, and also the outcomes that you're producing. So I think this is a big step, and there's a lot of work being done in this final step to smooth the way and to get a lot more of these trials through the regulatory steps. So these were kind of some main points that were surfacing through these various reports, and don't worry as well, I'll have if you haven't come across these reports or seen these reports, I'll have the full references in these slides, and you'll all be getting sent a copy of these slides following the webinar as well. So if you haven't seen them before, there will be the references and the links to easily find them following the webinar. So just quickly, just to run through so we're all on the same page, there are some main causes of missing data and then the implications of this. So as well, I just want to clarify what missing data is, because some people tend to have a different opinion on what it is. So when I talk about missing data, I mean this is information that you intended on gathering and for whatever reason a patient or a device or whatever broke down, or a patient withdrew from the study, so that this information that was intended on being gathered has not been collected. Some people have asked me before, when they set out and doing a study, halfway through or towards the end of the study they realize, well, geez, we should have really also collected another piece of information, and can we use imputation to fill in a reasonable or accurate value for that? And the answer essentially is no, because you're bordering on making up information then. This is really dealing with information that you intended on gathering and that wasn't collected for whatever reason. So different areas where the missing data can occur. I mean, it can crop up in issues with your trial design. It can crop up in issues with trial conduct. I mean, again, a lot of in these reports, the regulatory agencies and agents involved, a lot of them acknowledge that there is no real foolproof or airtight approach to defining and designing a trial. And I'm not going to preach about the pitfalls of trial design and conduct today, because that's another massive area and it's a whole other discussion to be had. What we're really focusing on, I suppose, is dropout rates here and issues to do with dropout, things that are completely out of your control. Sometimes things with trial design and conduct can be controlled and can be enhanced, whereas a lot of the issues with patient dropout and so on are just completely outside of any trial's control. And dropout rates can come into patients refusing to continue, patients withdrawing due to treatment failure or treatment success. You know, that they get better, they want to go home, they don't want to keep going back into a hospital or into a trial. Patients withdraw because of an adverse event that they had from the treatment, or simply a patient moves away to a different part of the country and it's just not possible for them to continue with the trial. So there's so many different possibilities and so many different ways that, of course, everybody accepts then that it's inevitable that you're going to get missing data, because it's so easy for it to pop up and so easy for it to occur, that you know, the main way then to or the obvious approach then is to kind of how to A, minimize it, and B, deal with what's left over. So I think the whole tackling it head on, because it is so prevalent, tackling it head on is a big issue, and also minimizing it can go a long way and can be the difference between a lot of trials being successful or not. So just to touch then on the impact of missing data, what are the knock-on effects of missing data cropping up in a trial. So some important ways that missing data can impact trials would be, obviously, as I mentioned before, introducing bias due to lack of a randomized sample. So some information that we were set on collecting, and it could be then a specific type of person has stopped or is having an adverse event or has left the study. A specific type of person has withdrawn from the trial, and therefore that type of person is not being studied anymore, and can be, and then this bias can creep in. Also, we had a specific sample size in mind, and because now we don't have these complete cases at the end of the study, we've got a loss in power, because the information we had initially planned on collecting is not there. We must include dropout cases in the data. So this is an interesting point. With the intent to treat principle, we're compelled to keep the information from every case in the data. But if you were to do a complete case analysis, those people get kicked out of the study, out of the statistical modeling or statistical analysis straight away. There are different methods of weighting and things to keep them in, but for the most part, they're not included in the actual analysis, the subsequent analysis. So this is an important point where multiple imputation can pick up on that, because imputation is certainly a way. Some people see it as, again, making up information, but I think the whole goal of multiple imputation and imputation as a whole is to come up with the most accurate value, not to answer that specific question, but to actually enable the analyst or the researcher to include the remaining information that had been already recorded. So the goal of imputation is to keep the information that has been recorded in the study, not to come up with answers for missing questions or missing data points. It's actually to include the information that has been already recorded. So that's a subtle point that I just wanted to make there. And also, I think we must figure out a way then to include the dropout cases in the analysis, and I think imputation goes a long way with that. The last point I make there on this slide is essentially there is data that we intended on collecting for analysis, and we're now missing it. So I think in terms of optimizing that approach, imputation is definitely one of the ways to go. So another point to come to here is the minimizing of missing data, and I think this is an important thing to focus on, and it definitely the National Research Council, in their report, the prevention and treatment of missing data in clinical trials, the first couple of chapters, they really push on this. Point that before you even get to addressing missing data, that you should really look hard at minimizing it first, because that can go a long way. There, in total, in this report, they make 18 recommendations, and recommendation number 2 to 8 are all based around reducing dropout rates through trial design, reducing dropout rates through trial conduct, and so on. So they do put a large emphasis on minimizing it. Before you concentrate on dealing with missing data, you should try and minimize it as far as possible. So some methods that they propose are run-in periods and enrichment before randomization, so that you have a good idea that the people you intend on including in the trial are committed to staying within the trial, and flexible dosing levels, so that you can accommodate for different individuals' ability to take the treatment, and also reduction in follow-up period, because it's quite known out there and quite well documented that there is an association with dropout rates and length of follow-up period. That with a longer follow-up period, it is harder to keep the large numbers in trials. Well, not large numbers, but the dropout rate can increase as the follow-up period increases. So it is important to try and minimize the missing data before we even consider tackling it. And a useful approach is to set a minimum level of completeness, is one thing that the National Research Council also recommend. So, and as I said before, to start this National Research Council report, these were recommendations that they're giving to the FDA when they were writing up, or as they're approaching and trying to tackle the issue of writing up on missing data analysis. So I think one thing now we can go to look at is actually getting ready for missing data analysis and getting your head around the missing data that is in your data. So once you've done your trial, once you've finished your trial and you have the data in front of you, you need to explore it first and just see the shape of the missing data, and is there any relationships between different variables and the missing data out there. So I'm just going to close out of this now and turn to Solus. So this is Solus here, and this is our statistical missing data tool. So Solus is oriented around imputation as a method for dealing with missing data. And again, it's a quite easy to use tool. We definitely engineered the interface around enabling not just statistical users but also clinicians and also I won't say non-quant users, but people who are not familiar with day-to-day coding of statistical algorithms and statistical models. So this is a very interface-based product. So I'm just going to open up a data set here. As you can see, it's quite in a kind of a spreadsheet format. And this is a very rudimentary example here just to give you the gist of it. So we've got four variables here, and we've got 500 cases roughly. We've recorded age of the patient, their BMI, hypertension, and cholesterol levels. And as you can see here, there's missing data dotted throughout the data set. So when exploring the missing data, the first thing you want to do is just to see the missing data pattern. And we have a drop-down menu here for this. So you go to View and Missing Data Pattern. And as you can see here, the interface is very drag and drop. So you just pick up the variables you want and you drop them into the boxes that you want them to go to. So I've just taken the variables into the variables to display box here. I'm going to click OK. And now we get a new window opened with a kind of a cell-by-cell breakdown of the data set. Here we can see that the blue dots, or the blue cells, represent missing data points and the white cells represent observed. And we can scroll down through this missing data view pattern to see the different data points. But if you have quite a large data set, this can be quite cumbersome, so we can collapse this down into a summary table, where across the top we have the 1, 2, 3, and 4, the variable names, and on the columns here we have the summarized cases or patients. So we can see here, for example, we have, of the 500, we have 358 fully observed cases. We have 72 cases with this particular missing pattern. We have 20 cases with this particular, and so on. So this is a very handy tool for just exploring initially the missing data pattern and how missing data has cropped up in the trial or the study. Another thing you can do here is split up the data into what we call a monotone or non-monotone missing pattern, and this is important for the next step, which I'll be talking about in our next webinar, which will be getting ready for imputation and getting ready for missing data analysis. So I'll just show you here briefly, but I won't get bogged down on this topic. So I've just now rearranged this table so that we can optimize for monotone and non-monotone patterns. As you can see here, the green is the non-monotone cells and red is monotone. And as you can see here, we've moved column 4 up to the second column. So essentially what we've done is we've just moved around the rows and columns so that we can create this maximized missingness pattern here and then this minimized missing pattern here. And what we do in Solus is we tackle these missing data points first, the non-monotone, so that we have as much information as possible to tackle these. Because you can see here that this data point has the least amount of information around it. Okay, but I'll be talking about this stuff in more detail at the next webinar when we're talking about looking at getting ready for imputation and missing data analysis. But essentially, this is the table that's quite interesting in just exploring the missing data and seeing how much have you observed and how much are you missing and where is it missing. We can also do some plotting. So in Solus we have various plots, various assumption checking as well as descriptive plotting. But we have this one plot here called a margin plot, which is slightly different, and essentially it's an expanded scatter plot. So I'll show you an example here. I'll plot BMI versus cholesterol, and again, as you can see, it's quite a very easy to use drag and drop system. You click OK, and what we've got here is essentially a scatter plot where the blue dots on the plot here are observed cases for both cholesterol and BMI. And then if you look down into the axes here or in the margins, you can see that we've got red dots. So on the x-axis here, this red dot signifies a value, observed value for cholesterol, but a missing corresponding value for BMI. And because there might be several red dots on top of each other, I can click on the actual individual values here. But because there may be several red dots on top of each other, what we've given here is just below that are two box plots. The red box plot illustrates the distribution of the missing data points, and the blue box plot illustrates the distribution of the observed. And this is quite a useful tool for just analyzing side by side the distributions of both missing and observed. We can also then modify this plot to, say if you wanted to break it down by a categorical or grouping variable. So in this data set, the age variable is a categorical variable where you got age is equal to 1, 2, and 3 different groups. And I can first, I'll just click OK. I can first split this up by different colors and symbols. So I've got blue circles, orange diamonds, and green squares for the different groups 1, 2, and 3. Or I can actually split this up by separate graphs. So then I can have 3 separate margin plots side by side. And what I'm doing here is I'm illustrating the different missing data relationships between BMI and cholesterol for the different age groups. So again, this is a very useful tool for analyzing the relationship between several variables and the missingness that has occurred. So these are very simple, easy to understand exploratory tools that you can use just simply by graphing alone in Solus before you tackle any missing data analysis. I think the final thing I just want to show you here as well is this is an imputed data set. I made 10 imputations, and at the bottom here you can see I've got 10 tabs, one for each data set. And these are now complete data sets. This is a different data set actually, by the way, but I just want to show you one thing is once you've actually done imputation, you can view, this is a scatter plot of 2 variables against each other. You can actually view the missing data points that you've imputed. So here we've got the red triangles are the imputed data points and blue are the observed, and I can actually overlay the 10 imputed data points on top of one graph. So again, this is a very easy to use visual display of the imputed data points once you've done imputation and missing data analysis. Okay, so that's just what I wanted to cover there in Solus in terms of exploratory missing data analysis. So that concludes the webinar for today, because I see we've just hit the 4 o'clock point there now, or the 12 o'clock in Eastern Time. So our next webinar in this series is Smart Choices for Imputation, or Smart Imputation Choices for Clinical Trials, and that's going to be on the 11th of April, so I welcome you all to come along to that. And the third and final webinar in this webinar series are Successful Strategies for Sensitivity Analysis, and that's on the 9th of May. So again, you're more than welcome to join us for that too. We will also be attending, Statistical Solutions will also be attending 2 upcoming conferences in the next few months. There's Society for Clinical Trials in Boston in May, and we'll also be at the Drug Information Association annual meeting in Boston as well, actually in June. So if you're intending on going to those conferences, please stop by at any time. So I'd like to thank you all again for attending our webinar, and I hope you found it hope you found it useful, and that you took some information from this. So I'd like to just open the floor there now for any incoming questions. We'll take a couple of minutes to take some questions there. So just give me 1 minute to consult there. Okay, so there was there's a nice few questions flowing in there as I was talking through the webinar. And thank you all for your input and for your questions. And please welcome your comments and questions as well. If you want to follow up in an email, my contact information is all here as well. So there was just one person asking a question on the imputation and the missing data pattern. So they were asking what was the difference between the monotone and non-monotone patterns. And essentially, when we want to set up the monotone pattern, we want to set up the pattern with most missingness around it. So every data point to the right is missing from the data point you're at. And then the non-monotone or the pattern is the missingness that we couldn't group together. And that's the missingness that you want to tackle first when imputing. Again, I'll be going into that in more detail in the next webinar when we actually tackle imputation and tackle it head on. Also, there was a couple of people asking, could they take a trial or try out their own data in Solus there, so they can do their own exploration analysis on their own data, and that's perfectly viable, right? If you go to solusmissingdata.com, you can download a trial, a free no strings attached trial, and it's fully supported. And if you want to read in your own data and play around with some visualizations and some imputations, that's fine. And again, it's fully supported, so if you have any questions when you're doing it, you can send us in an email, and we'll support you and help you get the most out of it. So again, running out of time there, so I better wrap up, but I'll get back to all the remaining questions personally by email, so I won't let anybody out. Also, we'll be posting a podcast of this webinar on YouTube, so again, if you want to look over it again, and if you have any further questions, please feel free to ask me as well. And also, everyone will be getting a copy of the slides from the webinar with the references and everything in there. So I'd like to just take the opportunity to thank everybody again for attending. We really appreciate it, and we're happy to answer any questions again that you have down the road if you want to go away and think about it first. So thanks everyone, and look forward to seeing you at the next webinar next month.